All right, it being a little after 1 o'clock and this being a very punctual place, we will get started. I'd like to welcome back, it's a pleasure to welcome back Ben Green, who's currently at the Australian Centre for Blood Diseases. Ben joined my lab 4 or so years ago as an honours student, and then when we were at the Murdoch Children's Research Institute, and then started his PhD there and completed it the last few months when we moved to the Walter and Eliza Hall Institute last year. So it's a pleasure to welcome Ben back and to tell us about some of the work he did during his PhD on the role of AKT in IL-3 signalling.
Thanks, Paul. So, as Paul mentioned, my project was focused on determining the role of AKT, specifically during IL-3 signalling. And the reason our lab was interested in IL-3 signalling was because even as far back as 2000 in the initial hallmarks paper, the evasion of apoptosis and the ability for cells to gain self-sufficiency from growth factor signals had been determined as critical points in promoting oncogenesis. And from downstream of the IL-3 receptor, a typical model by which this can occur is in the presence of ligand. Pathways such as AKT promote survival. When you lose a ligand, you deactivate AKT and you initiate apoptosis. And this is via the intrinsic or back dependent pathway. Typically what happens in leukemogenesis, for instance, may be the aberrant activation of AKT, and this promotes survival. However, this is independent of the presence of the ligand. So I'll briefly go over the BCL2 family proteins. The initial protein that was discovered was BCL2, and it was found to prevent cells from undergoing apoptosis. And of course, there's related proteins, including MCL1, which is found to be highly deregulated in numerous leukemias. There are also several related proteins, such as BAX and BAC, which were found to promote apoptosis, and indeed cytokine-dependent cells that are deleted of both BAX and BAC are unable to initiate apoptosis. These proteins share multiple homologous regions with the BCL2 family proteins, and there is a subclass of these pro-apoptotic proteins that only share a single homologous domain, the BH3 only domain. And the following is just a typical model by which these proteins regulate survival. So under normal circumstances, BAX-BAC initiates apoptosis, and the BCL2 family proteins function to promote, repress BAX and BAC and promote survival. The BH3 only proteins bind to and antagonize BCL2 activation, which again promotes apoptosis. There are other models that suggest that certain BH3 only proteins, such as BIM, can directly bind to BAX and BAC and promote apoptosis as well. So there are many different models by which the IL-3 receptor promotes survival, and the following is just one of those. And in this model, the IL-3 ligand causes the IL-3 receptor to promote recruitment of JAK2, and JAK2 is required for IL-3 survival signalling. And JAK2 activates multiple pathways, one of which is the phosphoinositide 3 kinase pathway, and PI3 kinase's role is to phosphorylate specific carbon residues on these phosphoinositol rings, or PIP rings. So it converts PIP2 into PIP3, and PIP3 acts as an anchorage point for AKT, and at the plasma membrane, AKT is activated. So the following is what is pretty much now the most dogmatically accepted model by which AKT promotes survival, and it stems from these two papers that were published in the mid-90s. And in this model, AKT's specific role was to phosphorylate the BH3 only protein BAD, and this meant that the chaperone protein 1433 now binds to BAD, translocates BAD away from BCL2, and it's then degraded by the proteasome. BCL2 then binds to BAX and BAC and inhibits apoptosis. However, if BAD does play such a central role in this pathway, why is it that cytokine-dependent cells that are deleted of BAD are still capable of undergoing apoptosis? And there have been several suggestions as to BH3-only proteins that replace BAD in this pathway, and they include PUMA, which has been shown to be required for normal apoptosis after cytokine deprivation, and also BIM, in that B cells that are deleted of BIM have increased longevity. So, as I mentioned, what we were curious as to how AKT was really promoting survival. However, I feel it's important to note that AKT is not the only way in which survival is regulated by the IL-3 receptor, and there's many different pathways that are all activated by IL-3 that all promote survival as well. And it kind of makes it a bit confusing to determine the specific role of AKT. Furthermore, there's this protein P10, and it's really been categorized much more recently than AKT was. And the understanding of PI3 kinase and P10's regulation of AKT is also important to understanding AKT's specific role. So as I said, PI3 kinase is responsible for phosphorylating these PIP3s. P10 is responsible for dephosphorylating them, and it acts as an antagonist of PI3 kinase. So a little bit more in depth on AKT itself. AKT has two domains. One is the kinase domain that possesses these two critical residues that when they are phosphorylated indicates AKT activity. And there's also this plextrin homology domain, and the pH domain is a negative regulator of AKT activity. And the way it works is that it folds over inactive AKT and hides these residues. When PIP3 is detected, AKT binds to PIP3, which means that, that AKT is brought into proximity of the activating kinases of AKT at the plasma membrane. Furthermore, by opening up and unfolding, these residues are now exposed for AKT activation. So there are 3 mammalian isoforms of AKT that vary in expression pattern, and they do have non-redundant roles when you look at the murine knockout phenotype. However, these are relatively mild phenotypes. And I'll also mention that none of these phenotypes particularly deregulate hematopoiesis. So AKT for a long time has been described as a very potent oncogene. And the reason for this was that AKT and more recently PI3 kinase expression has been implicated in a wide variety of cancers. And these include most commonly colorectal cancer and prostate cancer, but also glioblastomas, breast cancers, and in some circumstances even mutated in leukemias. And for a while, AKT has actually been used as a pathological marker of cancer. Furthermore, it has been suggested that P10 is the second most commonly mutated tumour suppressor behind P53, although this is a very bold statement. And this would suggest that the regulation of AKT is a critical step in oncogenesis. On the other hand, however, AKT-specific inhibitors have performed relatively poorly in clinical trials. And if AKT is such a potent oncogene, then where are all of the activating mutations? And it took until 2007 that this group, led by Carpton, identified an E17K mutation, which deregulates the plextrin homology domain. And they said that it immortalized certain cell lines. However, after this data was published in Science, there were numerous labs who replied saying that endogenous expression of this mutation is incapable of transforming cells, and these results were more likely an artifact of overexpression of this construct. And pretty much there is a lot of controversy surrounding whether this is a true activating oncogene. So this project was described in 3 basic questions. The first was, does AKT regulate survival in response to IL-3 signaling? And if it is capable of regulating survival, what is the mechanism by which it does so? And finally, if we can understand how AKT is regulating survival, can we determine that there are more effective methods of using AKT inhibitors in chemotherapy? So these are just 2 of the tools that we used. This is the 4-hydroxytamoxifen inducible lentiviral construct, which was modified to its present form by John Silk. And basically run off a ubiquitin promoter is this receptor transcription factor, GEV16. When 4-hydroxytamoxifen is present, the receptor activates the 5' UAS promoter and induces transcription of a gene of interest. And the genes that we were overexpressing in the construct were GFP, AKT1 and AKT2. However, these are heavily modified. So they both possess a hemagglutinin tag for identification. They're both myristoylated, which means that AKT is constitutively bound to the plasma membrane, so it's in proximity of its activating kinases. And furthermore, it's got a deletion of the plextrin homology domain, which was the negative regulatory domain of AKT. So I refer to this construct as hyperconstitutively active. For every single cell line that I'll show you that is expressing this construct, I would have performed FACS to determine the expression of hemagglutinin. These are 3 different back cell lines overexpressing GFP, AKT1 and AKT2, and they have been cultured in the presence or absence of 4-hydroxytamoxifen for 24 hours. And GFP overexpression does not increase hemagglutinin expression because there's no hemagglutinin tag. AKT1 or AKT2 overexpressing cells that are cultured in the presence of 4-hydroxytamoxifen have significantly increased expression of hemagglutinin compared to the uninduced cells, which is this dotted line. It's also important to mention that the dotted line appears to have increased hemagglutinin expression compared to the parental cell lines, which are grey, and this suggests that in the absence of 4-hydroxytamoxifen, there is still overexpression of AKT. And that's kind of important to remember for some of these results. On a molecular level, so these cells have been cultured in the presence or the absence of 4-hydroxytamoxifen for 24 hours before IL-3 was withdrawn at time 0. And as you can see, the induction of active phosphorylated AKT and total AKT is dependent on the presence of 4-hydroxytamoxifen, but not on the presence of IL-3 itself. Furthermore, there still appears to be some overexpression of the AKT construct which is independent of 4-hydroxytamoxifen, and you can also detect endogenous AKT in these Western blots. Given that the overexpression construct is so heavily modified, it actually runs at a higher molecular weight to endogenous AKT. The other important tool that we used were these factor-dependent myeloid cells, or I'll refer to them as FDMs. And these are a cytokine-dependent cell line that we feel is a good model of leukemia. And to generate these cells, you take an E14.5 murine liver, you co-culture these in the presence of fibroblasts that are expressing the HOXB8 retrovirus. You co-culture these in the presence of IL-3, and what you derive are a pre-myeloid progenitor cell. It sort of looks like this in its live version, and basically we consider these cells conditionally immortalized. So as long as you maintain exogenous IL-3 to the culture, these cells are happy to proliferate and survive indefinitely. When you withdraw IL-3 from the culture, even for 24 hours, the cells start to shrink, they bleb, and they stay in for annexin 5 and proprietary iodide. So we were very interested in how the IL-3 receptor was responsible for promoting survival, and we were partially inspired by Mark Guthridge and Angel Lopez, who had previously shown that the GM-CSF receptor has distinct survival and proliferative pathways that are varied by GM-CSF concentration. So in the presence of high amounts of GM-CSF, these cell lines were happily surviving and proliferating, and when you withdraw GM-CSF from these cells, they underwent apoptosis, and of course that means that they exited cell cycling. What Mark and Angel identified was a concentration of GM-CSF where you could promote these cells to maintain viability, however they were no longer cycling. And the hypothesis was that there was a distinct pathway at this concentration of GM-CSF that was being activated that was responsible for survival alone. Given that the GM-CSF receptor shares its signaling peptide with the IL-3 receptor, that is the beta common chain, we hypothesized that we could use wild-type IL-3 dependent cells in a similar manner. So just to point out, whenever I mention N in this talk, it will be the number of independent clones used, and all of the error bars are the standard error of the mean. So this was 8 independent wild-type clones that I then titrated into IL-3 and measured survival, which is the blue curve and the left-hand axis, and compared that to the percentage of cells that were in S phase, which is red on the right-hand axis. And these are just plotted from maximum to minimum. And in this case, even though there is a slight separation of these two processes, it's unclear whether you could actually culture these cells at a specific IL-3 concentration to maintain viability alone whilst the cells have exited S phase. So at the time, it didn't look like this model was working in our cells, but I took 4 of these cell lines and I plotted them separately, and in these 4 cell lines it appeared that apoptosis and cell cycle withdrawal were occurring at the exact same concentrations of IL-3. The other 4 cell lines, however, it looked like cells were exiting S phase at a significantly higher concentration of IL-3 compared to the concentration which they underwent apoptosis. So perhaps there was a concentration of IL-3 where we could maintain viability independently of proliferation. However, we could never identify a precise, robust concentration of IL-3 to do this in. So for now, we had kind of dropped this idea, although it will inspire us later on, as you'll see. So to look at the role of AKT in proliferation, these are now back-to-back double knockout cells that are overexpressing either GFP, AKT1 or AKT2. And in GFP overexpressing cells, when you induce GFP expression or for 24 hours before starving the cells of IL-3, in the presence of IL-3, you can see that there is no additive effect of GFP overexpression, and generally the cells are maintained at about 45 to 50% cell cycling. When you withdraw IL-3, these cells rapidly exit cell cycle and no longer divide. So if you were to induce AKT1 overexpression, in the presence of IL-3, there is no additive effect on the percentage of cells in S phase. If you withdraw IL-3, induced AKT1 overexpression seems to prolong the amount of time that these cells can be maintained in S phase. However, eventually these cells do exit S phase, which suggests that AKT1 overexpression is not sufficient to promote proliferation independently of IL-3. This was similar in AKT2 overexpressing cells. There's no additive effect of AKT2 overexpression, but there is a subtle, although significant, delay in the rate at which these cells exit S phase. So we had to look at this a different way, and these, being back-to-back double knockout cells, are capable of being starved of IL-3 without undergoing apoptosis. So we starved them of IL-3 for 24 hours before, at 0 hours, re-adding either IL-3, which is the solid lines, or 4-hydroxytamoxifen alone, which is the dotted lines. And as you can see, IL-3 rapidly reintroduced these cells into S phase, and these cells go on to continue proliferating. Unfortunately, AKT overexpression is not sufficient to promote these cells to re-enter S phase, and indeed these cells don't continue to proliferate in the absence of IL-3. So AKT overexpression is not sufficient to promote cell cycling or proliferation. So given that there was this supposed role of AKT in survival, we generated IL-3 dependent cells, these FDMs, that were derived from mice that were gene deleted for AKT1, AKT2 and AKT3. And even though there's no graph for this, this is a critical result in itself. That is that IL-3 dependent cells do not require each AKT isoform to promote IL-3 dependent survival. Nonetheless, we washed these cells of IL-3 and we stained for viability over a period of 5 days. And of course, back-to-back double knockout cells do not undergo IL-3 withdrawal-induced apoptosis. However, the deletion of each AKT isoform individually does not alter the apoptotic kinetics of these cells compared to wild-type cells. So, as I mentioned, we were inspired by this concept of multiple pathways being activated that were dependent on the concentration of cytokine that was present. So instead of doing a complete IL-3 starvation, we titrated IL-3 into these cells and tested them for the percentage of viability after 48 hours. And in this case, what we found was that AKT1 knockout cells were significantly less sensitive to low or suboptimal IL-3 concentrations compared to wild type or the 2 and 3 knockouts. So what this kind of suggests is that there might be a critical role of AKT1 in promoting survival somewhere here. And to quantify this, we measured the point at which 50% of these cells had undergone apoptosis. And it turns out to be almost a 10-fold difference. So we postulated that at this concentration here, the majority of AKT1 deleted cells were dead, but the wild-type cells were maintained in viability, and perhaps AKT1 is responsible for maintaining this viability. So we hypothesized that the opposite might be true. If we were to induce AKT overexpression, do we increase the sensitivity of these cells to low IL-3 concentrations? So these are wild-type cells overexpressing AKT1, AKT2, or GFP is the grey line. And in these circumstances, there seems to be a significantly increased viability of these cells grown in suboptimal IL-3 concentrations when either AKT1 or AKT2 is overexpressed compared to GFP. And it could be that AKT2 is capable of promoting survival by supraphysiological and forced overexpression of AKT2. So we tried to look at this one further way. We got hold of this AKT inhibitor, and this AKT inhibitor inhibits both AKT1 and AKT2. In 2 independent wild-type cells, cell lines, you can see that the AKT inhibitor is very potent at reducing AKT phosphorylation and therefore activity. So we took 3 independent wild-type clones. These aren't expressing the inducible AKT constructs. We cultured them in our normal high dose of IL-3, in the absence of IL-3, or at that suboptimal 0.02 nanograms per mil of IL-3. And to these cells, we added the AKT inhibitor. In the presence of IL-3, the AKT inhibitor is not particularly effective at inducing apoptosis, which suggests again that there are probably multiple pathways that are maintaining viability at this concentration of IL-3. In the absence of IL-3, the cells, of course, being wild-type cells, undergo apoptosis, but AKT inhibition seems to accelerate this. And one of the reasons this might be is that the AKT inhibitor is merely shutting down any AKT that has residual phosphorylation. The key result was here at the suboptimal IL-3 concentrations, where the majority of these cells remain alive. However, they turn out to be exquisitively sensitive to AKT inhibition-induced apoptosis. So again, it seems like the main role of AKT is to promote viability at this concentration of IL-3. So if I was to draw up a model of how IL-3 was signaling, in abundant IL-3, there appears to be multiple redundant pathways that promote both survival and proliferation. And thanks to Anissa, we knew that in the absence of IL-3, P53 was required to promote transcription of puma, and this was one of the ways in which these cells underwent apoptosis. The data that we had accumulated so far suggested that there was a third pathway that was activated in only low IL-3 concentrations, which activated AKT1 alone, and this was somehow capable of promoting survival. And we were really curious as to how this was working. And we had plenty of evidence in the literature that suggested it was through the regulation of the BH3-only proteins. So we took multiple wild-type cell lines and infected them with the inducible AKT1, AKT2, or GFP overexpressing constructs. We cultured these cells in the presence of 4-hydroxytamoxifen for 24 hours before starving the cells, and we looked at viability over a period of 5 days. And in wild-type cells, AKT1 or AKT2 overexpression is sufficient to prolong viability after IL-3 deprivation. However, supraphysiological overexpression of hyperconstitutively active AKT is not sufficient to promote, to maintain viability independently of IL-3. And again, it points to the fact that AKT is not critical for regulating survival in the absence of IL-3. In the absence of both backs and back, obviously these cells no longer undergo apoptosis. So we reasoned that if you were to delete a gene that was critically regulated by AKT to prolong survival, then when you deleted that gene and you overexpressed AKT, you would no longer see an effect of AKT overexpression. And to start with, we took the bad knockout cells, and the reason we chose this cell line was because it was the central protein in which was theorized to be critical for AKT to regulate survival. However, even in the absence of bad, there appears to be a significant prolongation of viability when either AKT1 or AKT2 is overexpressed. And this suggests that AKT overexpression does not require bad to maintain viability in the absence of IL-3. We did the same thing this time in BIM knockout cells, and again it seems like AKT1 and AKT2 induced overexpression is sufficient to prolong viability after IL-3 deprivation. So BIM is also not required for AKT overexpression induced survival. And then we did the same experiment in puma knockout cells, and this was quite surprising because when we overexpressed AKT2 in the absence of puma, there was an increase in viability that prolonged survival after IL-3 deprivation. However, in AKT1 overexpressing cells, there no longer appears to be any effect of AKT1 overexpression to promote survival. So this data suggests that AKT1 at least requires puma to promote survival after IL-3 deprivation. So for every single data point here, the cells were also taken out of their media that was starved of IL-3. They were replated back into IL-3 and also soft agar, and we simply counted the number of colonies that formed. and this is referred to as the clonogenic potential of the cells. And we measured the clonogenic potential of the cells due to AKT1, AKT2 overexpression compared to GFP overexpression. And again, in wild-type cells, there seems to be a clonogenic advantage to having overexpressed AKT1 or 2. And given that back-to-back double knockout cells do not lose clonogenic potential due to IL-3 starvation, there's obviously no effect of increasing AKT overexpression. Now, just watch out because the axes here have changed, but in bad knockout cells, induced AKT1 or AKT2 overexpression significantly increased the clonogenic potential of these cells after IL-3 deprivation. Similarly, in BIM knockout cells, there is a significant increase in clonogenic advantage due to AKT1 or AKT2. Then we got to the Puma knockout cells, and again, AKT1 overexpressing cells do not have an increase in clonogenic potential in the absence of Puma. In AKT2 overexpressing cells, there was only a single time point at which there was a significant difference. So this data suggests again that AKT1 is regulating Puma to promote clonogenic advantage. One of the questions we'll commonly ask though is, at 2 and 3 days these cells are undergoing apoptosis, how do we know that's not because we've deregulated the AKT inducible construct due to prolonged IL-3 deprivation? Given in wild-type cells the majority of these are now dead, it was hard to do to probe for AKT overexpression, so we used back-to-back double knockout cells. These are 2 independent clones. They were induced for AKT for 24 hours and then starved of IL-3, and then lysates were generated at 48 and 72 hours. And 4-hydroxytamoxifen is still capable of maintaining AKT overexpression independently of IL-3. So this suggests the reason these cells are dying is simply because AKT is not sufficient to prolong survival at these time points. So on a molecular level, Jared took the wild-type cell lines, he cultured them in the presence or the absence of 4-hydroxytamoxifen and then starved them of IL-3 for up to 24 hours. And when probed with Puma, these cells seem to increase in Puma expression in GFP overexpressing cells by 4 hours, but the majority of Puma is degraded by 24 hours as the cells begin to undergo apoptosis. AKT overexpression does not prevent the buildup of Puma, however it does appear to delay Puma expression after IL-3 withdrawal. And as I mentioned earlier, just because there's no 4-HT in the culture doesn't mean that these cells are not overexpressing AKT. And this was similar as to what was seen in AKT2 overexpressing cells. And one reason for this is that even if AKT1 was requiring Puma to maintain viability, there were Puma-independent pathways that AKT2 was capable of regulating, but it still appears that supraphysiological overexpression of AKT2 is sufficient to delay Puma expression. Jared also probed these lysates for MCL1, and in GFP overexpressing cells, you lose MCL1 expression as IL-3 is starved. But what happened was it appeared that AKT overexpression maintained MCL1 after prolonged IL-3 deprivation. And this was interesting to us because Ulrich Maurer and Doug Green had suggested that AKT was specifically regulating MCL1 to maintain survival. And the model that they used was that AKT phosphorylates and therefore inhibits glycogen synthase kinase, which would otherwise repress MCL1 expression. However, before we were willing to accept this model, we were curious as to whether these cells had increased MCL1 because AKT was specifically regulating MCL1 to maintain viability, or whether these cells had increased MCL1 because AKT was maintaining viability, which indirectly allowed MCL1 expression. So we repeated this, but this time in back-to-back double knockout cells, and these are only AKT1 overexpressing cells, again cultured under the same conditions in the presence or absence of 4-HT for 24 hours before IL-3 was starved. Again, in these cells, you see that there is a delay in expression of Puma when you induce AKT overexpression compared to GFP overexpressing cells. And again, going back to this role of BAD being critically regulated by AKT by phosphorylation and proteasomal targeting, well, indeed, induced AKT overexpression seems to phosphorylate BAD, but if anything, it seems BAD levels are increased due to AKT overexpression. So this again suggests that AKT is not specifically regulating BAD to maintain survival. To show you this model again, AKT phosphorylates GSK3, which would otherwise inhibit MCL1 expression. So again, phospho-AKT correlates with phosphorylated GSK. However, this increase in GSK does not maintain MCL1 expression after prolonged IL-3 deprivation compared to the uninduced or the GFP overexpressing cells. So it doesn't appear that AKT is specifically regulating MCL1s for survival either. So Dimi then took the wild-type cells, cultured them in the absence or the presence of 4-hydroxytamoxifen for 24 hours before starving the cells of IL-3 for the indicated amount of time, and then probed them for Puma expression and MCL1 expression by qRT-PCR. And I'll focus on Puma first. GFP overexpressing cells have an increase in Puma expression after IL-3 withdrawal, and obviously this is independent of GFP overexpression. Again, just watch the axes because they change here, but it seems that cells that possess either the AKT1 or AKT2 construct have reduced expression of Puma, but furthermore, when you induce AKT overexpression, there is further reduced expression of Puma after IL-3 deprivation. So it seems that induced AKT overexpression is repressing Puma in response to IL-3 deprivation. When you look at MCL1 expression, however, GFP obviously does not affect MCL1 maintenance after IL-3 deprivation. And then when you add AKT1 or AKT2, there, if anything, appears to be a decrease in MCL1 expression, not an increase, which is what we'd expect if AKT was promoting survival. So again, this suggests MCL1 is not specifically regulated by AKT. So as we fill in this model again, when AKT is responsible for promoting survival, it does so through repressing Puma. And again, we're interested in how this occurs. And given this was occurring via a transcriptional mechanism, we hypothesized that there may be transcription factors that are being regulated by AKT to repress Puma after IL-3 deprivation. So two candidate genes were p53 and FOXO3A, and the reason for this was that both genes are regulated by cytokines, both genes are promoters of Puma transcription, and so we generated a similar setup, p53 and FOXO3A knockout cells possessing the AKT1, AKT2 or GFP overexpressing constructs, induced AKT overexpression and starved the cells for 5 days of IL-3. And again, in wild-type cells, you see prolongation of survival due to AKT overexpression, and even though it's subtle, there is still prolongation of survival in the absence of FOXO3A. What was critical was in p53 knockout cells, induced AKT1 overexpression has no effect on prolonging viability when either AKT1 or AKT2 is overexpressed. So this suggests that both AKT isoforms require p53 to regulate survival. Again, we did the clonogenicity of these cells at each time point. Again, the axis here goes down a little more. However, wild-type cells overexpressing AKT1 or AKT2 have significantly increased clonogenic potential after IL-3 deprivation. FOXO3A knockout cells, unfortunately, none of these time points are significant, and so we would suggest that maybe these cells require FOXO3A for clonogenic advantage. However, if you compare these to the p53 knockout cells overexpressing AKT, where there is absolutely no increase in clonogenicity due to AKT overexpression. So both AKT1 and AKT2 appear to require p53 to promote survival. So these are multiple cell lines from the indicated genotypes, wild type, Puma, bad. They were cultured in the presence or the absence of p53, and to these cells the AKT inhibitor was added and the cells were tested for viability. And the reasoning behind this experiment was to see whether AKT inhibition-induced apoptosis required any of these genes. So you've seen the wild-type data previously, but basically in the presence of IL-3, the AKT inhibitor is not particularly effective at inducing apoptosis. And this is true for all of the genotypes tested. So we'll focus on the cells cultured in the absence of IL-3. And in wild-type cells, again, it turns out these cells are sensitive to AKT inhibition-induced apoptosis. So are bad and FOXO3A knockout cells. And this would suggest that you do not require bad or FOXO3A to induce apoptosis, which is caused by AKT inhibition. But surprisingly, it turns out to be true for Puma as well. So if AKT1 requires PUMA to promote survival, why is it that inhibition of AKT does not require PUMA? And the reason for this may possibly be due to the nature of this AKT inhibitor in that it inhibits both AKT1 and AKT2. And as I showed, there were PUMA-independent mechanisms by which AKT2 overexpression maintained viability. The critical result, however, was that p53 knockout cells are not undergoing apoptosis caused by AKT inhibition. And in fact, if you take 4 of the p53 knockout cells and 4 of the wild-type cells after they've been starved for 24 hours and you replate them back into cultures with abundant IL-3 and soft agar, it turns out that the AKT inhibitor is capable of repressing clonogenicity, even in the presence of IL-3. In the absence of IL-3, of course, you see a decrease in the clonogenicity as well due to AKT inhibition. It also turns out that p53 knockout cells are susceptible to a loss of clonogenicity due to AKT inhibition, even in the presence of IL-3. And this means 2 things. The first is that clonogenicity is not being maintained by AKT via p53-dependent pathways, but also the AKT inhibitor is still capable of affecting p53 knockout cells to begin with. So again, this model suggests that when AKT is responsible for survival, it does so by repressing p53, which would otherwise transcribe PUMA. And again, we were curious as to how this was occurring, but under these circumstances, we had a pretty big clue, and the reason was because the literature was replete with references to AKT's ability to regulate MDM2. So MDM2, typically in its phosphorylated form, is fairly stable and it binds to and inhibits p53. Unphosphorylated, it's rapidly targeted by the proteasome. So Rachel Lee took some wild-type FDMs and cultured them in the absence of IL-3 for 4 hours before re-stimulating them with IL-3 for 20 minutes. And there is a robust increase in phosphorylation of MDM2, which correlates to AKT activity. So I took these one step further, and this time I took back-to-back double knockout cells that express the AKT1 inducible overexpression construct, starved them of IL-3 for 24 hours before re-stimulated them with IL-3 alone or 4-hydroxytamoxifen alone. And under these circumstances, IL-3 is sufficient to promote phosphorylation of MDM2 and increase the total expression of MDM2. 4-hydroxytamoxifen alone, and thereby AKT1 overexpression, is sufficient to promote phosphorylation of MDM2 and stabilize the total amount of MDM2 present. So our model of how IL-3 is regulating AKT to promote survival looks a little like this. In abundant IL-3, multiple redundant pathways promote both survival and proliferation. And in the absence of IL-3, p53 is responsible for transcribing PUMA to promote apoptosis. In low concentrations of IL-3, it appears that AKT is primarily responsible for survival by phosphorylating MDM2 and thereby allowing MDM2 to inhibit p53. When you lose IL-3, you lose AKT activity, and MDM2 is now proteasomally degraded, and this results in the freeing up of p53. So in conclusion, to redress those questions that we initially asked, does AKT regulate survival in response to IL-3 signaling? And there are both pros and cons. On one hand, given that you can generate AKT knockout-dependent IL-3-dependent cells, AKT is not required to promote survival due to IL-3 signaling. Furthermore, even if you supraphysiologically overexpress hyperconstitutively active AKT, you still cannot promote promote survival in the absence of IL-3. On the other hand, specifically AKT1 seems to be required for survival at certain contexts, such as suboptimal IL-3 concentrations. So when AKT is responsible for survival, it appears to be doing so via a p53-dependent mechanism, and there are at least both PUMA-dependent and independent mechanisms of survival as well. There is another conclusion, however, and that is that survival signaling by AKT1 is completely dependent on PUMA. However, because we've supraphysiologically overexpressed AKT2, it's also capable of repressing PUMA as an off-target effect and promoting PUMA-independent pathways to maintain survival. So finally, Does this mean now that we have a greater understanding of AKT's mechanism of survival, can we more effectively use AKT inhibitors as chemotherapeutic drugs? So I'd like to finish with a bit of a story, and this was an experiment that until 2 weeks ago I had kind of assumed had failed. And basically I was trying to maintain cells in these suboptimal IL-3 doses by inducing overexpression of AKT1. And what I expected was that AKT1 would maintain viability for significant amounts of time at these low IL-3 concentrations. So these are two GFP overexpressing clones and 3 AKT1 overexpressing clones cultured at this concentration of IL-3 and the presence of 4-hydroxytamoxifen, and they were just tested for viability. And GFP overexpressing clones died before 2 weeks had finished. Unfortunately, 2 of the 3 AKT1 overexpressing clones also died. So AKT1 was not sufficient to prevent these cells from dying. What was curious was that there was this one clone that did maintain viability right up to a month after I had initially put these cells into this low IL-3 dose. And at first I assumed that this was due to AKT overexpression. So I washed the cells of 4-hydroxytamoxifen to see if now these cells would suddenly crash. And they didn't. So AKT overexpression is not required for these cells to have viability, and therefore these cells aren't transformed by AKT. To see if this was reproducible, I took the same clone again, did the same conditions again, and this time it underwent apoptosis at a very similar rate at which the initial 2 clones did. So at first I thought this didn't work. However, what it does provide is insight into the period that AKT is responsible for transformation of cells. Out at this prolonged IL-3 deprivation, these cells are no longer dependent on AKT, which means that they are no longer, they would no longer be susceptible to AKT inhibition. There seems to be, at this point here, a cell that has accumulated numerous mutations, therefore, that have allowed this cell line to survive in this low IL-3 concentration. So perhaps what's happening is that here there is multiple mutations that have been accumulated that is due to the prolonged survival of this cell line caused by AKT overexpression compared to GFP. And this suggests that AKT inhibitors aren't effective at this long term in the absence or in the low concentrations of IL-3, but there is a very brief period at which these cells require AKT to promote transformation. And I'll leave it up to a clinician to decide whether it's feasible to target AKT at such a small window of opportunity. So I have to thank lots of people who were involved in this project, particularly Paul, Anissa and Rick, who were the supervisors for this work, but everyone in the lab of course helps out, and I really need to thank everyone as well. I also worked a significant amount of time over at Peter Mac, where I shared Rick Pearson's lab. And I also got cells from Karen Montgomery and Vanessa Solomon and Brian Hemmings. Furthermore, a lot of people had input into this project who weren't at WeHi or the Children's, and they include Mark Guthridge, John Silk, Andrew Way. And whilst I was at the Children's, I have to thank Matt Burton especially for his help on facts, but also Elizabeth Alga and David.
Plenty of time for questions.
Just 2 quick questions. Did you look at cell lines like in both Puma and Vim?
No.
And then the second cell line was that resistant cell line mutant for P53?
I did not check. So unfortunately that data I kind of wrote off because it didn't really fit in with anything I had. But only recently have we really started discussing what happened.
Nick.
I apologize if you said this early on because I walked in a little bit late, but you pointed out the difference between the effects of high or low concentrations on the different pathways that become critical. High, low concentration of IL-3. Have you worked out, what the sort of equilibrium constants or whatever for those pathways are, and how they drop, why they drop away?
No, so I'm trying purposely to avoid designating which pathway is active because there are 2 possible explanations as to what's going on. The first is that AKT has the lowest equilibrium point and is activated by the lowest concentration of IL-3, and it really is just a matter of titrating IL-3 in until only AKT is active. The second is that there are multiple pathways that lie downstream of the IL-3 receptor, and that AKT is playing a critical role at that concentration of IL-3 that is only signalled by the receptor under certain circumstances. But I don't know whether one of those is more feasible than the other.
Oh, I feel like a Melbourne auctioneer. Are you all done?
Is there a role for P10? I know that you avoid the 3 effects by your constructs, but could P10 concentrations be being modulated?
So we didn't actually look at whether P10 was being regulated by AKT, but P10 would not be affecting the overexpression of AKT. And the reason for that is that being a myristylated construct, it's irrelevant whether PIP3 is being phosphorylated because the AKT construct will automatically be targeted to the plasma membrane. Whether AKT has a feedback loop that promotes or represses P10 is actually one of those topics that a lot of people in the PI3 kinase field are trying to understand now. And it does appear in the literature at least that there may be mechanisms by which AKT represses P10 activity.
Lee?
You looked at FOXO3 in detail and didn't see much happening. Did you consider the expression or the activity of any of the other FOXO family members like 1 and 4?
No. So there was a reason we chose FOXO3A, and that was because we had previously seen in the literature that FOXO3A was being specifically regulated by IL-3, and also that it was a potent promoter of Puma expression. If we were to test many different members of the FOXO3A family, unfortunately we'd have to generate those cell lines as well, and it seemed like a lot of work when the FOXO3A knockout cells being the most likely candidate weren't having any effect on AKT-induced survival.
If I'm not mistaken, this AKT is also downstream of the insulin receptor, and there's a story that insulin resistance drives certain cancers, particularly bowel and other solid tumours. Have you thought about whether you might be able to see if insulin modulates the effect you see? Is there any insulin, for instance, in the medium that you're currently using, and have you thought about whether you might be able to tease that out a bit further with the system?
Yeah, I mean, your question actually touches on another point, and that is whether there are isoform-specific effects of AKT. So, for example, AKT2 is known to have critical regulatory abilities downstream of the insulin receptor, and in fact, the phenotype of the AKT2 knockout is that they tend to be hypoglycemic, and we didn't look for this under these circumstances because we were more interested in the IL-3 dependent pathways. But it is true that AKT has an acknowledged role in maintaining viability and even glucose metabolism downstream of the insulin receptors.
The short answer is no, we haven't looked.
Yeah, sorry.
I'm going to bother.
No, well, I don't know. I mean, we just started seeing what glucose deprivation does to these cells in the presence or absence of AKT, which is not very much difference. We don't know what the insulin receptor levels are on the surface of the essentially myeloid progenitor cells.
And you presumably have a little bit of insulin in you've got serum containing.
Serum, yeah.
But you probably don't have very much.
I think the other answer to that would be, though, however, that these cells are absolutely dependent on IL-3. So any of the effects that we do observe are because of IL-3 deprivation and not necessarily because of background insulin.
You might have to modulate that with insulin.
So I guess I've always had kind of a slight problem with the survival with low doses of IL-3 because I'm not. I mean, what do you think that means? Presumably it means it's whatever it was, 0.02 nanograms per mil at some very, very low level of receptor occupancy.
Mm-hmm.
You've saturated your survival responsibility to work on P53 and Pure and so forth. Why would that saturate? I mean, why would activating more receptors just recruit more?
Yeah, so as I said, there are two models by which this would work. The first would be that there are specific pathways that are regulated by certain concentrations of IL-3. The second is that that at low concentrations of IL-3, the only pathways that are being phosphorylated or activated due to a KD being significantly lower in AKT than any of the other pathways that I showed. Rick Pearson had a suggestion that perhaps what's happening is not that AKT1 is critical for viability, but that the majority of AKT in these cells is actually AKT1. So by knocking out AKT1, you actually lose the majority of the survival signal that is being propagated by AKT in total. And therefore these cells are now more sensitive to low IL-3 concentrations, as there's just less AKT to promote that signal.
All right, well, if that's the finish of all the questions, we could just thank Dean for the seminar.